Good afternoon, students. Good afternoon, everyone. Whether I'm audible? Good afternoon, everyone. Good afternoon. Yes. Yes, good afternoon. Whether I'm audible? Yes, yes. Good afternoon. Good afternoon, Udaya. Good afternoon, Mani. Okay. Morning session. Yes, morning session. We have completed first 60 question from previous year question paper discussion of 2023. Okay, shift 1. Okay. Now we are going to see the remaining 60 questions. Okay. Kindly be attentive. Okay. We are going to discuss. Each and every important points. Okay. What are the questions? Okay. They have asked. What is the explanation? Okay. Okay. Then what are the relative objectives that can be asked from the similar portions? Okay. Then most important thing I want to say before starting this session, from our last 5 to 6 sessions, we are completely discussing about this previous year question paper only. If you see, some of the core subject, like some of the core topics are there, questions are repeatedly asking from that portion. Okay. While listening the class, you have to take the notes also. Okay. These are the core area they are covering repeatedly. So that you have to focus much for your preparation. Okay. Clear? Now you have to answer. Okay. First question. Which of the following is multiple sequence alignment tool? This they asked in 2024 also. Which will be the answer, student? Which of the following is a multiple sequence alignment tool? Whether scope or PubMed, PDB, Gold or Cluster W. What will be the multiple sequence alignment tool? Any guesses? This they have asked in 2024. Match the following they have asked. Any guess which will be the multiple sequence alignment tool? Whether it will be a scope or PDB or Gold? Yes. Cluster W, excellent. Okay, this we have already seen there. Okay, SCP-OP means what? Structural classification of proteins. Okay, here structural classification of proteins will be there. This is protein data bank. Okay, these are some other different tools. So the tool that is specifically used for multiple sequence alignment is Cluster W. This is one of the repeated question from bioinformatics tools. Okay. Second, what is the first sequenced protein? They are asking what is the first sequenced protein. This is very similar. This we have seen in 2024 question also in match the following. Sanger did sequencing of insulin. Okay, he has done the first insulin. Okay, insulin he has sequenced first. Yes, correct. Insulin. Okay. Now we are going to some of the important points related to other options. Okay, hemoglobin. They will ask this question. Okay, hemoglobin. Who has discovered this one? Hemoglobin. Okay, this they can ask. Then who discovered myoglobin? Okay, who discovered hemoglobin? Who discovered myoglobin? Then most important thing, what is means? This myoglobin is completely made up of alpha helix. 8 alpha helix will be there. This they can ask. Then another important thing, hemoglobin will have alpha 2 and beta 2 structure. Okay. Heterotetramer. Okay. Heterotetramer. This is a heterotetramer. Okay. This question they can ask. Then another important question what they can ask from here is hemoglobin will show allosteric activity will be there. Okay. Phenomenon of cooperativity will be there in hemoglobin, but myoglobin is a monomeric protein. There will be a no allosteric regulation. So these are the key points. Okay. Golden rice is a variety was developed by whom? This we have discussed in genetic engineering class. Golden rice was developed by which scientist? Yes, option B. Ingo Potix and Peter Bayer. Golden rice. Okay. That means vitamin A rich or beta carotene rich rice. Okay. Here we also have to focus about the different genes. We have already seen in our class what are the different genes they have taken. From maize they have taken, then from daffodils they have taken. That also we have to focus. Okay. So the question is golden rice, who has discovered? The scientist name here writing here. Okay. Lacopron. See, every year they are asking one question from Opron. Lacopron or tryptophan opron. Read the question. Lacopron contains 3 genes. Lac ZYA. These genes are transcribed as a single mRNA under control of one promoter. Yes, that's why we are saying that is a operon. Operon means multiple genes will be controlled under a single gene. Yes, A is correct. When lactose is available, the lac repressor bind tightly to the operator. They are saying if lactose is available, the repressor will go and bind with the operator region, preventing the transcription by RNA polymerase. Whether this is right or wrong? They are saying if lactose is there means that will be repressed. No. If lactose is there means this will be activated. Correct. So that will be the wrong answer. So A is correct, B is wrong. So wherever B is coming, that is wrong. Okay, we can eliminate here itself. Then C, operator overlaps with the promoter and it is a negative regulator site. This is correct. Yes. Next, lac operon contains the genes that specify proteins to cell to utilize the lactose. That is, we know lac ZYA genes are there. This will code specific enzymes that will catabolize the lactose for uptake into the cell. So this is also correct. So the right option is what? A, C and D. Yes, very good. Enzyme required for removing RNA primer. Enzyme required for removing RNA primer during DNA replication. During DNA replication, which enzyme will be utilized for removing the RNA primer? Removing the RNA primer. Yes. Yes. DNA polymerase 1. What is this command bottom of 1? I'm not getting. DNA polymerase 1. Option D. What will be the answer? Yes. So DNA polymerase 1 will be the right answer. Why? Because this DNA polymerase will have 5' to 3' exonuclease activity. This will be involved in removing of what? It will be involving in removal of the primer after replication. Yes, very good students. Sigma factor is a component of which polymerase? Whether it's a component in DNA polymerase 1, 2, 3 or RNA polymerase or endonuclease, which will be the component? Yes, sigma factor is a component in RNA polymerase, DNA polymerase or endonuclease. Yes, excellent. RNA polymerase. Okay, we know the sigma factor will have this. Sigma factor is a component in our RNA polymerase. Apart from that, we have alpha to beta, beta dash, omega and sigma. This is the entire component of RNA polymerase. This beta subunit will have the catalytic activity. Okay. This sigma will be involved in promoter recognition. Okay, promoter recognition. Clear? Yes. Here they have given different crops. Then some of the mutated genes they have done. Because of mutation, they are getting some benefits. Okay. They have done genome editing here. Okay. For example, maize. They have done something genome edited thing. Because of that, what trait can be improved? So maize, ultimately it will come waxy starch. Okay, this for commercial application we can do. Okay, for industrial application. Soybean, specifically protein and oil rich crop. 2 will come D, 1 for 3. Potato, browning is the major problem. So, non-browning variety can be done. Okay, so 3 will come 2. Tomato will have high level of GABA. Why means they have enhanced the activity of this GABA biosynthetic enzymes in tomato fruit. Okay. So 3, 4, 2, 1. Where is coming? 3, 4, 2, 1. So option D will be the right option. Yes, 3, 4, 2, 1 will be the right option. Again, RNA polymerase that is required for synthesis of mRNA. Which RNA polymerase is required for synthesis of messenger RNA? Yes, which one? RNA polymerase 1, 3 or RNA polymerase 2 or reverse transcriptase? Which will be required? RNA polymerase 1 or 2 or 3? Yes, RNA polymerase 3. RNA polymerase 2, yes. RNA polymerase 2 is involved for mRNA synthesis. Okay. RNA polymerase 3 will be involved for tRNA synthesis along with 5S ribosomal subunit. Okay. This is very important. 5S ribosomal subunit. RNA polymerase 1 for ribosomal RNA. Apart from that, that means 5-8, 18S and 28S. This is most important thing. So, which RNA polymerase will be involved for mRNA synthesis? Means RNA polymerase 2. This question they have asked specifically for eukaryotics. Okay. Eukaryotics. In prokaryotics, single RNA polymerase, a single RNA polymerase will transcribe all the genes. Clear? So, a single RNA polymerase will transcribe all the things. Okay. Type 2 restriction endonuclease are the most abundant enzyme used for gene cloning. Type 2 restriction endonuclease includes, which is the example for type 2 restriction endonuclease? They are asking, which is the example for type 2 restriction endonuclease? Whether EcoR1, yes, excellent. EcoR1, okay, EcoR1 is a typical example for type 2 restriction endonuclease. In 2024, they asked, EcoR1 will produce what type of ends? It will produce 5' overhang head ends. Clear? In 2019, they have asked, what is the restriction site for this E. coli R1 TK? That is what? 5' GAATTC. TK, this is the restriction site. TK, recognition site. This they have asked. TK, so see, from same option, different type of set they have question. Different set of questions they have said. Okay, so you have to learn about the options also, where it will come, what it will do, like that. Okay, that's why we are discussing this previous year question paper. We are not going for only one option. We are discussing what it will come for other things also. Okay, 5' end of DNA is characterized by it. If a DNA is there, what will be there at the 5' end? What will be there? Whether it will have hydroxy group, peptide bond, nitrous group, or it will have phosphate group. What it will have? Read the question properly. Yes. Read the question properly. Then answer. Yes. D. Excellent. Okay. 3' hydroxy will have. 3' will have hydroxy group. This will go and bring a nucleophilic attack on 5' phosphate present in DNTPs. Okay. So 5' will have phosphates group. Okay. Next, they have given the different techniques. Again, technique in biochemistry, more than 3 to 4 questions they have asked. They have given different techniques and what is its application. Okay. Southern blotting, this we know. Southern blotting, for which technique we will use? Southern blotting, PCR, ELISA, gel electrophoresis. Among them, simplest technique is PCR. PCR, why? For what purpose we will use? For the amplification of the DNA fragment. So, D will come to. D will come to. Where coming D is to? Okay, D will come to. Then gel electrophoresis. Gel electrophoresis, what we can do? We can separate the DNA based on size and charge. So 3 will come, 3 only. So this is the right option. Yes. 4132. Yes. 4132 is the right option. Yes. 4132. Option B is the right option. Excellent. Then we can see remaining things. ELISA. There we will use what? Enzyme linked immunosorbent assay. We will use some of the enzymes that will convert colorless product. Okay, colorless substrate to product. Okay, for example, horseradish peroxidase we can use, like this enzymes we will use. Okay. Then southern blotting. This is simple thing. What southern blotting? What it will do? Transfer of DNA fragment from electrophoric gel to nitrosulfurous sheet. This is very easy. Another thing, plant hormone question. This is very easy question for everyone. 2 hormones that are required for callus to differentiate. Callus to differentiate. What are the 2 hormones? What are the 2 hormones that are required for callus to differentiate? Yes. Auxin and cytokinin. Repeated question. If high auxin is there, means root will form. If high cytokinin to auxin ratio is there, means shoot will form. If equal ratio of auxin and cytokinin is there, means callus will form. This is also another important thing. So the two hormones required for callus differentiation is auxin and cytokinin. Institute. In 2023, they have asked two institute. First, they asked National Institute for Plant Biotechnology is located at where? New Delhi. Second thing they have asked, National Bureau of Agricultural Important Microorganism is located at where? National Bureau of Agricultural Important Microorganism is located at where? Chandigarh or New Delhi, Mau, Mumbai? Yes, excellent. Mau. Mau, UP. Okay, clear. So this is clear from institute. There is high possibility of having one question in general agri. Okay. So you have to read what are the institute, major institute, and what are the and where it is located. Okay. What are the institute and where it is located? That too is very important point. Okay. Next, hybridoma technology. Hybridoma technology is generally used for production of. This will be used for production of antibodies. That is clear. Whether it is a monoclonal antibodies we will produce or polyclonal. Yes, excellent. We will produce monoclonal antibodies in hybridoma technology. Yes, Uday Lakshmi, excellent. Manohar, excellent. Okay. So, monoclonal antibodies will be produced by hybridoma techniques. This is one of the most important technique. Which of the following vector can carry the longest piece of foreign DNA? Among them, which will carry the longest piece of foreign DNA? Whether bacterial artificial chromosome or YS? Yes, yes. Yeast artificial chromosome will carry large number of fragments. Okay. Large number of foreign insert DNA can be carried. Okay. Followed by bacterial artificial chromosome, approximately 300 kb. Okay, this will be greater than 1000 kb we can carry. What is the size for plasmid and plasmid? DNA insert size for plasmid and plasmid. Plasmid, it will come around 8 to 10 kb we can clone. Plasmid, approximately around 45 kb we can clone. Okay, this is also important point. So, YAC is what? High capacity vector. Okay, we can clone a large fragment. It's a high capacity vector. Reverse transcriptase enzyme is used for what purpose? Reverse transcriptase enzyme is used for what purpose? Repeated question. Reverse transcriptase enzyme will be used for mRNA synthesis, tRNA synthesis, cDNA synthesis, and vector synthesis. Yes, it will be used for cDNA synthesis. We will isolate the mRNA. We will treat with reverse transcriptase enzyme for formation of cDNA. This cDNA, this is what? First strand synthesis of cDNA. Yes, this is first strand synthesis of cDNA. For second strand synthesis, there are two ways are there. One is replacement synthesis, another is self-priming. Okay, here we will use RNase-H. This they asked in 2025. We have already discussed this question. Okay, so reverse transcriptase will be used for cDNA synthesis. Who has discovered that one? Temin and Baltimore. This is also important question. Okay, Temin and Baltimore. Temin and Baltimore has discovered this one. Okay. Which of the following is a vector mediated gene transfer? From different gene transfer method they are asking. Which of the following is a vector mediated gene transfer method? Biolistic or agrobacter mediated, gene gun or microprojectile? Yes, agrobacterium is what? It's a vector based gene transfer method. We can use certain virus also, plant viruses like cauliflower mosaic virus, Gemini virus, TMV. This also we can use. Okay, these all are vector mediated. Okay, physical or chemical transformation, there this will come, biolistic, gene gun, micro injection, everything will come. Okay, clear? Yes. Some base substitution. Read the question properly. Some base substitution do not result in change in amino acid sequence. If a base is substituted, okay, that is not changing the amino acid sequence of polypeptide. Why? Because of what property of codon? Whether the university of the codon triplet nature of the codon, or collinearity, or degeneracy of genetic codon. What will be the right option? Yes, degeneracy of genetic codon. We know a single amino acid can be coded by more than one codons. A single amino acid by more than one codons. This is due to degeneracy of genetic codon. We know arginine, leucine and serine will have 6 codons. 6 codons will code for this each amino acid. Methionine and tryptophan will have only single codon. This they asked in 2020. 2020. Tryptophan codon is UGG. This is very important. Tryptophan codon is UGG. Single codon. AUG, this is start codon, you know. AUG. Methionine will be coded. Next, they have given Book's theories or purport characteristics. Like what is meant by ortholog, splicing, mass spectroscopy, different tool they have given, EMBL tool. Then they are asking to match the question. Among them, what will be the easy? Splicing, mass spectroscopy, these are one of the common terms. Splicing, RNA splicing. Specifically, we will use for removal of introns. So, 2 will come 1. Yes. Second is option 1. Where is coming second option 1? Only here is coming. Simple thing. Yes. 2 is coming option 1. So, orthologs means what? Homologous genes found in different organism. Yes. Excellent. 4. Then mass spectroscopy, protein fingerprinting, EMBL. That will be a nucleotide database. This is one of the simple question. Okay. Next important thing, genetics. They are asking about Mendelian genetics. Okay. The T1 plant show 3:1 segregation. Observe here, T1 plant is showing 3:1 segregation for selected trait and the gene. Okay. When the 3 T1 plants with the target genes were selfed, they are taking a T1 plants and they are selfing together. Which of the following statement explains the result? Let me know what will be the correct answer. They are saying which of the following statement explains the correct crossing. Let me know, simple. Whether all the 3 plants will show the same progeny with the genes, or 2 of the 3 plants produce the progeny plants with the gene, or only 1 plant out of the 3 plants produce all the plants with the gene, or all the 3 plants will have the gene. Yes. Example. Excellent. Tejaswini, excellent. Only 1 out of the 3 plants produced will have the progeny with that particular gene. This is a simple crossing. Okay. This is simple mathematics only. You will work out. Okay. The intracellular space, plant physiology question. The intracellular space between cell membrane. They are asking cell is there. Okay. What is the intracellular space between the cell membrane and the space of the cell wall is termed as, yes, middle lamella. Yeah, the intracellular space between the cell membranes and the space out of the cell wall is termed as, yes, excellent, middle lamella. Excellent. Okay. See here, post-harvest physiology. One question, this everyone can write post-harvest physiology question. See here, statement. They have given multiple statement. Yes. Almost all post-harvest technology manipulate the metabolism of the harvested highly perishable produce by minimizing the respiration. Yes, they can minimize the respiration and increasing the water loss. Whether they will increase, no, they have to decrease the water loss. Okay, so this will not become, yes. Respiration is a post-harvest issue. In post-harvest tissue, often increase the temperature of the perishable produce during storage. Yes, it's an exothermic process. Respiration is a exothermic process. Heat will be produced. So ultimately it will increase. So B will be the correct. Wherever B is coming, that will come in the answer. Physiological maturity. 2024 they asked this one. Okay. Physiological maturity refers to stage in the development of fruits and vegetable when maximum growth and maturation have been occurred. This definition they have asked in 2024. Okay. So this is correct. So where B and C is coming? B and C is coming here. Oh, in all the option it's coming. high transpiration rate from the harvested produce will not lead to any economical effect. No, it will cause an economical effect. So wherever D is coming, it will not come. So B and C will be the right option. Clear, students? So B and C will be the right option. Yes. The highly perishable farm produce. Which will be the highly perishable farm produce? Apple, tomato, cauliflower, finger millet. Yes, very good. Tomato is one of the perishable farm product. Okay. Tomato. We can use blanching to decrease the perishability. Okay. This will decrease the enzymatic activity in cauliflower. This we know. Okay. Another thing from electron transport chain. Okay. The component of the plant mitochondrial electron transport chain that provides an alternative route for electron passing through the electron transport chain. We know four complexes will be there. There is a component. This will passes the electron directly to oxygen. What is that component name? Whether it is cytochrome C oxidase? Yes, excellent. Alternate oxidase. Okay, alternate oxidase. In 2024, they asked where this alternate oxidase will be located. It will be located in mitochondrial membrane, not in chloroplast. It will present in mitochondrial membrane. Okay, clear? This we can also say as cyanide resistant respiration. Why? Means the cyanide will inhibit the complex 4. Okay. This alternate oxidase directly transfer the electron to oxygen. So cyanide resistant respiration can be happened. Okay. The nutrient element that is considered as mobile element in plant. Mineral nutrition. The nutrient element that is considered as mobile element in plant. What is the mobile element in the plant? Yes, magnesium. What are the immobile elements? See, immobile element, calcium, sulfur, boron, iron. Okay, these all are some of the immobile element. Okay, these are some of the immobile element. Apart from that, see, phosphorus is typical element that will be immobile in soil, but that will be mobile in plant. So you have to read the question properly. If they are asking about phosphorus mineral nutrient, whether they are asking the mobility nature of this element in soil or plant, then only you have to work out the answer. Okay, so the right option for this is magnesium. Clear? Yes. Next thing, the biochemical reaction in which carbohydrates are converted into aromatic amino acids. Simple. Carbohydrates, we know this will give carbon skeleton for biosynthesis of amino acid. They are asking which is the specific pathway that will be involved in synthesis of aromatic amino acid. Any guesses? This we have, yes. Cyclic acid pathway. This we have already discussed in our class. What will be the inhibitor for this cyclic acid pathway? This also I have already told, practical application. What will be the specific inhibitor? The structural analog of PEP. It is an herbicide. That also I am saying. It's an herbicide. What will be the answer? Glyphosate or glufosinate? What will be the answer? Glyphosate or glufosinate? Yes. Glyphosate or glufosinate. Which will be the answer? That will specifically inhibit the EPSPS enzyme. EPSP enzyme. Yes, glyphosate. Okay. Glyphosate is a herbicide. This will compete with PEP. It will go and bind with this EPSP enzyme. Because of this, aromatic amino acid biosynthesis will not be there. Plant will be toxic and it will be leads to death. Okay. The commonly translocated compound, that means the photosynthesis in the phloem is what? Whether it will be sucrose or mannose or fructose or glucose, what will be there? Yes, sucrose. Why sucrose is transporting, not other sugars? Why sucrose only is not, it's transporting? We know alpha, beta, 1 to 2 will be there from glucose and fructose. Okay, this is important linkage. Okay, why? Why sucrose is transporting? The most important thing is, this is another important thing for your understanding. I am saying, okay, sucrose is non-reducing in nature. That's why it is transporting in phloem. Okay. Another important thing, if glucose is transporting, this will increase the osmolarity. This will increase the osmolarity of the cell sap. Okay, because this is reducing, so it may increase the osmolarity. That's why cell has wisely chosen sucrose as a transporting molecule for carrying the sugars from one place to other. Okay, so sucrose is the right answer. Okay, choose the wrongly matched answer. Sorghum, Kranz anatomy. Yes, it's a C4 plant. Kranz anatomy will be there. Blackman law of limiting factor. This is correct only. Yes. IBA is a natural auxin. Yes. PS1 is P680. Whether PS1 is P680? Yes. D only is the. Then PS1 is what? 680, 690, 700, 720, 730. What will be the answer? PS1. Yes. P is 700. Yes. Very good. So 680 is PS2. Excellent. Next, strigolactons. During our phytohormone class itself, I told, from strigolactons, sure they will ask one question. See, we are getting the question. Strigolactone, a natural plant growth hormone, has been shown to regulate the major role. What it will do? Whether it will regulate the root inhibition or branching or flowering or fruit set. This in class itself I have told. Three major function will be done by strigolactone. Think properly based on the option. Think properly, take your time. Yes. The most important thing is branching will be regulated by strigolactone. How means? How it will regulate the branching? It will inhibit the branching. Okay, by inhibiting branching. Okay. Root initiation is general work. Okay. So much of hormones will play that root initiation work. For example, jasmonic acid also will initiate the root. But the specific for strigolactone is branching will be regulated. Branching pattern will be regulated. And specific for brasinosteroid, if you see what it will do, leaf angle will be regulated. Clear? Clear? Students, clear? Yes. So the specific for strigolactone is branching. These questions only students will going to make a wrong answer. Okay. So you have to read and grasp the question properly, then you answer. Okay. So the right option is branching. Okay. Then here they have given same question from plant hormones. They have given the different inhibitors. Then they are asking to matches. Okay. Match TIBA. TIBA. It's an what? It's an antioxidant. This we know. It's an antioxidant or auxin inhibitor. TIBA, where it is? It's an inhibitor of auxin. So, 1 will come option 4. Where is coming? 1 option 4. Okay, 2 things it's coming. MCP. MCP is an what? It's an ethylene inhibitor. This we already seen. Yes. MCP is an ethylene inhibitor. 2. Okay. Already answer came 4, 3, 4, 2, 3, 1. Yes, we can solve. Paclobutrazole. Paclobutrazole is specific for what? Flowering in mango or regulation of leaf angle? Yes, flowering in mango. Okay. Brasinosteroid, earlier only I told brasinosteroid, it will regulate the leaf angle. So 4, 2, 3, 1. 4, 2, 3, 1. So option B is the right option. Yes, very good students. The formative effect of IAA. Listen the question properly, read properly, then you answer. Okay. Formative effect of indole acetic acid. They are asking about the specific thing. Formative effective of indole acetic acid. Whether it will induction of bud dormancy will be there, maintenance of apical dominance, induction of senescence, or prevention of cell division. Yes. What will be the right option? maintenance of apical dominance. It's the critical role of this auxin. Yes. The head rot of beets, stem crack of celery or water core of turnip are the deficiency symptom of which mineral nutrient? From mineral nutrient, they have asked 2 questions so far. Okay, 2 questions they have asked. Yes, excellent. Boron will be the typical symptom. Boron will cause this typical symptoms: head rot of beets, stem crack of celery, and water core. Okay. From mineral nutrition, so far we have seen 2 questions they have asked. Okay. Next, the typical earliest symptom of magnesium deficiency in plant. We know, we know magnesium is what? It's an mobile element. Yes, it's a mobile element. So what it will do? It's a mobile element means it will mobile in plant. So deficiency will always happen in older leaves. This is clear. So deficiency will always open in older leaves. This is the right option, intravenous chlorosis of older leaves. So 3 questions from mineral nutrition they have asked this year. Next, they have given different crops. We have to match with what is the example for each and every crop, whether it is a C3 crop, C4 or CAM or C3, C4 intermediate. Okay. Just match and let me know what will be the answer. Go with the easy thing. Pineapple, yes, we know it's a CAM plant. So 3 will come option 4, where 3 is coming option 4. Okay. Then alternaria. Alternaria, it's a typical example for C4. Yes, D will come option 3. Here D is coming option 3. Rice is a C3 plant. Yes, option 1. So C will be the right answer. 1, 2, 4, 3. Yes. 1, 2, 4, 3. Yes, excellent. 1, 2, 4, 3. Rice is a typical example for C3, C4 intermediate. Can anyone tell? 1, 2, 4, 3. Yes. Now it's clear. Yeah. Then what will be the answer? 1, 2. Just rice major C3. No. Yes. Rice is a major example for C3 crop. So first option will come 1 only. This is correct only. Here option is wrong, students. Okay. You see here properly. Rice is a major C3 crop. Pineapple is a CAM crop. Alternaria is a C4. This is the intermediate between C3 and C4. Okay. Here typing mistake is there. Just use C1. Okay. Next. Yeah. If you want to practice PYQs by topic wise, or for section wise, you can kindly enroll in this one. Then you practice as much as possible. Practicing PYQ is one of the major thing for clearing the exams. Practice well. Those who are targeting for ICAR 2027, they can join in our classes. For first 100 students, there is offer is going on. Kindly enroll as soon as possible. Next, retention of full genetic potential for the development into a complete plant is what? What? Retention of full genetic potential for the development into a complete plant. A single cell is will be developed into an entire plant. This is what? Retention of genetic potential in a differentiated cell for the development into a complete plant is. Whether it's a redifferentiation, dedifferentiation, totipotency, what will be the answer, students? Totipotency means what? Yes, totipotency means the individual cell that will have a capability to develop into whole plant. Don't be confused. Okay, they are asking something, they are twisting the words and they are asking. Same definition only. Totipotency means that can develop into a complete plant. Redifferentiation. Their complete plant will not come. First, mature cell can be converted into callus cell. This callus can be converted into the entire plant. Okay, clear? Uday Lakshmi, clear? So, yeah. So, you shouldn't be confused between redifferentiation and totipotency. Yes, done, done. Next, the scientists who proposed that chemical messengers are responsible for growth and development of plants. For growth and development of plants, chemical messengers are required. Which scientist has proposed this one? Boyson and Jensen or Julius Van Sachs, Skoog or Darwin? Boyson and Jensen. Boyson and Jensen, I think he has given this water cycle, Benson oxidation cycle, water oxidation cycle. This we can also say as oxidation of water. Julius Van Sachs. Okay, Julius Van Sachs, water is essential for growth. Okay, water is essential for growth of the plants. That has been given by Julius Van Sachs. Chemical messengers are responsible for growth and development of plant. Who has given? Huh? Who has given? Any guesses? Darwin? No. Yes, you have to write the answer now. Yes. Whether it is Falcon Skoog or Darwin or Julius Van Sachs or Boyson Jensen. You have to answer. I will wait up to that. You have to answer now. What will be the right answer for that? Julius Van Sachs will come or these things? Who? Boyson and Jensen or Skoog? Skoog, specific for kinetin and zeatin. Okay. The right answer is Julius Van Sachs. Okay. He is only proposed these two terms. Okay. Clear? In this year, they have asked two scientist names. Okay. Majorly two scientist names they have asked. Okay. Phytohormones are photoreceptors required for light sensing in plant. The red lighting absorbing pigment, PR, wavelength is what? What is the wavelength for PR and what is the wavelength for PFR? Yes, let me know what is the wavelength for PR and what is the wavelength for PFR. PR and PFR. Yes. Yes. Why? What happened? PR and PFR. This is very easy question. PR means what it will come? 667. PFR means 730. Yes. 667 will be the answer. Yes. Yes, they are correct. See, again, growth parameters. Okay, growth parameters. They are asking about leaf area index. 2025, they have asked the same question, leaf area index. Leaf area index means what? Total leaf area of the plant to the ground area occupied by the plant. This is another important thing is, this is unitless. Okay, unitless. This we have seen. Yes, option A is the right option. Water use efficiency. 2025, 2024, 2023. 3 years repeated question, this one, water use efficiency and transpiration rate. Okay, 3 years repeated question. Water use efficiency means what? Amount of carbon dioxide assimilated as biomass or amount of carbon assimilated per unit amount of light. Light will not come, this will not come. This is what? Harvest index. What will be the answer? Yes, amount of carbon assimilated as biomass or grain produced per amount of water used. Okay, very good. The phenomenon of cold requirement for flowering is termed as starification, vernalization, photoperiodism, and thigmotrophism. What will be the answer? Stratification or vernalization, photoperiodism? Yes, vernalization. Vernalization. Cold requirement for flowering. Yes, vernalization is the right option. Then where this stratification will come? Stratification also cold treatment we will do. But for what purpose? For breaking dormancy. Correct. Yeah, for breaking seed dormancy. Yes, clear. In actively growing plants, like for example, rice, wheat, groundnut, etc. is there, the component of water potential become negligible. They have asked the same question in 2024. They have given 1 meter plant height is there. What are the components are not involved for calculating the water potential? We know water potential is solid potential, pressure, gravity, and metric potential. Typically, yes, C and D. C and D will not come. Okay, which is not, see, this gravitational potential and metric potential we will not use for calculating the water potential for this crop. So they are asking the question, this component of potential become negligible. So negligible will be what? C and D. Yes, very good. Nucleolus is the site for synthesis of this component. In nucleolus, what will be synthesized? Whether ribosome will be synthesized, DNA or chromatin or nucleosome, what will be synthesis? Yes, ribosomes will be synthesized specifically in the nucleolus. Okay, ribosomal, in ribosomes, specifically the ribosomal RNAs will be synthesized. Okay. An example of long day plants repeated. Every year they are asking one example from whether short day, long day or day neutral. Then another thing, they are asking example for C3, C4 on camp plant, example for then orthodox seeds, recalcitrant seeds. Okay, this is repeated question. So you have to focus much on that. Okay, what is the example of long day plant? Yes, cotton is a short day, chrysanthemum short day, tobacco is also short day. So sugar beet is the critical example for long day plant. Another important thing, they are asking from photosynthesis. This we have discussed in our class itself. Okay. Statement 1 they have given, DCMU, also known as diuron, is a herbicide, is an inhibitor of light reaction of photosynthesis. Statement 2 they have given, DCMU acts by accepting electron from early receptor of PS1. What will be the right option? Yes, this I have discussed in our class itself, lecture itself. What will be the answer? I already told paraquat, paraquat, diquat. This paraquat is also known as methyl vilogen. Methyl vilogen. These are the typical herbicides. This will be what? It will inhibit PS1. DCMU. This were PS2. So this is wrong statement. So the right thing is what? Statement 1 is correct and statement 2 is incorrect. Yes. Option 3 is the right. Chlorophyll biosynthesis begins with which amino acid? Chlorophyll biosynthesis begins with which amino acid? This I told in class. Which amino acid? Chlorophyll biosynthesis. Whether it's glycine, glutamic acid, aspartic acid or alanine. Excellent. Glutamic acid. Glycine and succinyl-CoA. Okay. This will come for glycine and succinyl-CoA will come for animals. In plants, glutamic acid. Okay. Glutamic acid is the specific precursor for heme biosynthesis. Okay. Photorespiration is found to be zero. They are saying photorespiration will be zero or negligible in this crop. So photorespiration will be zero for camp plants, it will be zero because daytime it is closing the stomata. Then what? C4 plants photorespiration will be zero. Why? Kranz anatomy is there that will prevent the photorespiration. C3 plants will have more photorespiration. In our question, they are asking photorespiration is found to be zero or negligible for what are the crops. We have to just wisely, we have to choose C4 plants and camp plants. Okay. What will be the answer? Yes, pineapple will come. Yes. Amaranthus is a C4 plant. Yes, it will come. Then anything else? Finger millet. Sunflower. Yes, very good. Very good option. D. Yes, very good. Okay. Next thing, quantum yield of oxygen production. They are asking here quantum yield of oxygen production in light reaction of photosynthesis. Light reaction of photosynthesis. First, we have to know what is meant by quantum yield and quantum requirement. Okay. Quantum yield means the name itself is there. It's a quantum. Okay. Quantum yield means number of oxygen molecule. See, we can simply say oxygen molecule release for each molecule of oxygen release, how much number of photons are there. Okay. Quantum requirement means number of photons that are required, to release a molecule of oxygen. A molecule of oxygen. Okay. This is quantum requirement. Requirement means number of photons required. Quantum yield means number of oxygen molecule released per photon. Clear? This is clear. So they are asking about here quantum yield of oxygen production. Okay. Quantum yield means what? So oxygen released per number of photons utilized. Okay. Around 8 to 10 photons are required to release. Exactly, if you say around 8 photons are required to release a molecule of oxygen, 1 divided by 8 is 0.125. So approximately it will be 0.1. Excellent. Uday Lakshmi, 0.1 you have mentioned. Yes. 1 by 8 equal to 0.15. Yes, very good. Identify the scientist who gave the term hydroponics for growing of plant in water culture. They are saying who is the scientist given the term hydroponics. They are not asking about any nutrient medium. Yes, very good. Very good, very good. Okay. Gerrick is the scientist. He has proposed the term hydroponics. This Ebuta and Sumuka are what? They have prepared different nutrient mediums, hydroponic mediums they have prepared. Okay. So you have to choose the option wisely. Okay. This is another important thing they have asked about post-harvest physiology. So post-harvest physiology, everything they are asking 1 or 2 questions. Okay. They are asking easy questions only from post-harvest physiology. Okay. See here, with reference to waste life of cut flowers, identify the correct statement. Okay. For prolonging life and quality, cut flowers are often held in holding or waste solution. This will be correct. Yes, we have to increase the shelf life. Okay. The waste solution contains a combination of carbohydrates, growth regulators, inhibitors, and minerals. Third, any waste solution must contain essentially 2 components, mineral salts and ethylene inhibitors. No, whether all the waste solutions should have, no, it's not mandatory to have the ethylene inhibitor. Whatever the flowers that are sensitive to this petal senescence, that means ethylene mediated petal senescence will be there. Na? Whatever flowers are sensitive, for that only we will use the ethylene inhibitors in the waste solution. So C will never come. Okay. A will come. Okay. Mineral salts helps in preventing plugging of conducting tissue, while ethylene inhibitors make available the respiratory substrate. Whether ethylene inhibitors will available make the substrate, no, carbohydrates only, na. So this also wrong. Okay, this also wrong. So what will be the right answer? A and B will be the right answer. Yes, A and B will be the right answer. Seed dormancy is broken by mechanical scarification in this crop. In which crop the seed dormancy will be broken by mechanical scarification? Which crop? Whether cotton, castor, cotton we will use delinting. We will use acid here. Sulfuric acid, concentrated sulfuric acid we will use. Yes, excellent. Castor. Okay, castor, in castor, seed dormancy will be broken by mechanical scarification. Okay. Okay, clear. Identify the orthodox seed. See, they are asking example of orthodox seed. Orthodox seed example. First, we have to know what is meant by orthodox seed. It's a desiccation tolerant seed. Okay. It's a desiccation tolerant. Recalcitrant. Another term is the recalcitrant. This is desiccation intolerant. Okay, desiccation tolerant and desiccation intolerant. So what will be the right option? They are asking about orthodox seeds. Identify the orthodox seeds. Example, what will be the example? Sorghum and cotton or mango and yes, very good. Okay, sorghum is the typical example. What about cotton? What about cotton? Hmm? Cotton also example of orthodox seeds only, na? Okay, both A and D. Okay, read question properly. Okay. Again, about photosynthesis only they are asking this question. Yes, cotton also. Yes, very good. Cotton also will come as agriculture orthodox. Okay. So last one option A and D is the correct answer. Okay. Now they are asking two statement they have given about light compensation point. Okay. The light compensation point will be low for C4 plants. This we know. LCP will be high for C3 plants. If you know this basic concept, this question will be easy. The photosynthetic photon flux density at which CO2 uptake by photosynthesis exactly equal to carbohydrate released during respiratory process is called light compensation point. Yes, this is correct. LCP of sun plants range from 1 to 5 picomicromole per second, whereas corresponding value for shade loving plants is high. They are saying shade loving plants will have high. Whether this is true? No. Shade loving plants will have low, sun loving plants will have high. So statement 2 will be wrong. Okay. So statement 1 is right and statement 2 is incorrect. I hope this is clear for everyone. Okay. The enzyme requiring nickel as a cofactor in higher plants. What enzyme requires nickel as a cofactor? Nickel as a cofactor. Which enzyme requires nickel as a cofactor? Alkaline phosphatase, carbonic anhydrase, zinc it's required. Yes, what will be the answer? Catalase, iron will be required. This will require magnesium. What will be the answer? Yes. Urease, nickel is typical for urease. TK. TK. Nickel is a cofactor for urease enzyme. TK. Don't forget, this is important. TK. Statistics question. From last 4 years question paper discussion, we have seen only one statistic question. TK. We have seen most of the general agri question from genetic portions. TK. Because that is there in our plant biotechnology syllabus also. We have seen there more question. Then we have seen about institutes and its location. From there also we have got so much of general agri questions. TK. So see here statistics, the test which is used to determine the difference. They are saying the difference between observed frequency and expected frequency. This is simple thing. Which test will be used to determine the difference between observed frequency and expected frequency in one or more than one category? Yes, excellent. Chi-square test we will use. Yes. Same 2024 they have asked the same question. In 2023 also same question they have asked in part 1. Okay. What different crops they have given and major state of production. Cotton, where will be the major production? Mustard. Mustard means Rajasthan, this we know. Then potato, UP. Red gram, Karnataka. Cotton, Maharashtra. Clear? Clear. This one is clear. So 4, 3, 1, 2. This is very easy question only. I know you will answer. Okay. 4, 3, 2, 1. Okay. They have given this while taking our class itself. I told they can ask what are the biomolecule synthesis and what is the energy source that will be utilized, whether it is using ADP or CDP or AMP. This I told in our class itself. Sucrose biosynthesis will happen in where? Cytoplasm. Yes, it will use UTP glucose. TK, UTP will be utilized there. TK. So A, wherever fourth option is coming, that is the correct. TK. Nitrite reduction that will require FADH2, fatty acid synthesis, NADPH. This is a biosynthetic process. Substrate level phosphorylation, GTP formation in TCA cycle. Okay. Next, identify the crops based on amount of water consumed per gram of CO2 fixed. They are asking about water use efficiency only. Indirectly, they are asking water use efficiency will be more for which crop? We know C3 plants will C3, C4 and CAM. CAM will have the highest water use efficiency, followed by C4, followed by C3. Just to match here. So this will be the right option. Yes, option C will be the right. Pineapple will have higher water use efficiency, followed by maize, followed by rice. Okay. Next, identify the nutrients. See, fourth question from mineral nutrition only. Identify the nutrient element in crop plants capable of getting redistributed. That means it is if a plant is about to die or if a leaf is supposed to senesce, the nutrient in this leaf has to be relocated or redistributed to other parts of the plants. If it is redistributed means first important criteria that nutrient should be mobile. Correct? They are asking indirect question, capable of getting redistributed under deficiency condition in descending order. So whatever is mobile, highest mobile, okay, that will only will mobile first. Okay. So boron not mobile, sulfur not mobile, phosphorus, okay, calcium not mobile, here also not mobile, here also not mobile. So nitrogen is highly mobile, iron is partially mobile, boron is highly immobile. So this will be the right option. Clear? This will be mobile. Yes. Option D is the right option. Yes, very good, Udaya. Identify the synthetic inhibitor used in crop plants. What is the synthetic inhibitor used in crop plants? Whether malic hydroside, chloramate chloride, paclobutrazole, triiodobenzoic acid or triiodobenzoic acid. What are the example of synthetic inhibitors used in crop plants? Malic hydroside, chloramate chloride, paclobutrazole, triiodobenzoic acid. What will be the right option, students? Yes. What will be the right answer? A and C or A and D, B, C, D or A, B, D? What it will come? A, B, C. Benzoic acid, triiodobenzoic acid. What will be the answer? Yes, very good, very good, very good. Okay. This is the last question. Then this is the repeated question. This is the repeated question. Okay. They have asked 2 times. Okay. They have asked 2 times the same question. Most commonly used chemical to break dormancy requiring light in seeds like oats, lettuce and gladiolus are. What are the components, chemical components they will use for breaking the dormancy in oat, lettuce and gladiolus? This is repeated question. Yes, I need answer from you guys. What will be the answer? Whether potassium nitrate, kinetin, thiourea or NaA? Yes, thiourea, potassium nitrate, kinetin and NaA. Excellent. A and C. Potassium nitrate and thiourea is the common chemicals that are used for breaking the dormancy. Okay, breaking the dormancy requiring in the seeds. Okay, clear. Very good. Any doubt is there? If no doubt is there means we can end the session today. Okay, tomorrow we will see 2023 shift 2 question paper. Okay, any doubts are there? Kindly reply me whether these videos are useful or whether I have to improve something. Kindly let me know that also. Okay. Take care, students. I will see you in the next class. Thank you.